Hello and welcome to the tutorial of I3CR-WANO. In this video, I will show you how to use I3CR-WANO using XNAT. In a different video, I have talked about how to use I3CR-WANO using Docker, but in this video, I will restrict myself to just the XNAT usage. Please note that all the code, documentation and pre-trained models for this framework can be found at these two GitHub repositories. Now let's look at the GitHub repository and the documentation. So if I go down in the README file, I can see a field which says running with XNAT. And so within running with XNAT, I have all the details and instructions for running I3CR-WANO within XNAT and how to set it up. And we can follow along by following that GitHub repository. But now I am in an XNAT instance where I go to the images and commands option and I pull my two Docker containers. The first one is near new Ronco preproc and it has this 7 different commands associated with it. And then I pull my second image, which is energy new Ronco segment, which has a single command associated with it. Note that it will take a little bit time to pull these commands, but here for the sake of this video I have sped it up. Now once the commands are pulled, we go to command configurations and enable all these commands site-wide. And then if you want to use it in a specific project, you go to this project, which in this case is I3CR-WANO demo, and we enable these commands within the project. So we have to do two enabling, one is site-wide and then one is project-wide. Now looking at a sample session within this I3CR-WANO demo project, I see the option of running these different containers within the session context. And so then what I do is, the first thing I do is I go to custom variables and I create a custom variable called segmentation ready, which will show me if my session has all the sequences required for segmentation or not. If it does not have this, then this flag gets set to false, and if it has all the required sequences, then it is set to true. So now we go to the sample session and you can see that this field here is now empty, but when I run the scan type classifier, it will be set to either true or false. You can see this series class column which is empty now, but once I run the scan type classifier, it will get filled up with the different scan types. Now I run the first command, scan type classifier, and it starts running. You can check the status of this command from this window below, and you can see the standard output log, and I can see that the container is running now. So the container is running, and here I have sped it up for the sake of this video, and once the container stops running, it will show that processing completed, just like it shows here. And I exit this standard output log, and if I reload this box, I see that my running has completed. And now you have to reload the page to actually see the fields and the series class column. So when you go and reload the page, we see that the segmentation ready field is true and the different series the scan types are set. You can go to manage files and see that a folder called one scan type classifier is created, which has the different scans in patient space and the CSV file containing the scan types. Now I am running the second command, which is registration, and just like before, I go down below and reload the page to see that it has started running, and I can check the standard output log from this window. So first, the scans are co-registered to the patient space and then they are registered to a standard Atlas space. The registration has stopped running now. I reload the page and go to manage files, and I see in the registration folder I have the co-registered scans and I have the QC file to quickly check if they're okay or not. And I also have the registered folder which contains the scan registered to a standard template, and I see that they all look good. So now I move on to the third command, which is skull stripping, and it goes the same way as before. I reload the page, I can check the output log, and I wait for it to get over. So I see that it is running now. processing is completed, I exit this window, reload the page, and I go to manage files to check my scans. So for this one also I have a QC file which shows me quickly if the scans look good or not. Then I move on to the segmentation step. I run the container and the segmentation takes place. So my segmentation is now complete, and I quickly go and check the segmentation quality by checking the QC file for this one as well. So in this case it is a seg.png file where I have the different slices and the tumor shown in the bottom. This is a multi-class tumor segmentation, so the red is the necrotic or non-enhancing core of the tumor, the yellow is the enhancing core, and the green is the edema. And things look good, so I move on to the prediction to patient space command, which takes this segmentation and converts it back to patient space from the atlas space. Note that this step and all the following steps are pretty quick. So this one is already done, and it also creates an output folder under resources, which is the 5 segmentation patient space folder. And with that I move on to the patient space to DICOM-SEG format, where I basically take the nifty file and convert it into DICOM segmentation object format. Now this is also done, and then the final step, which is a step required only for XNAT, is where I take this patient space prediction in DICOM-SEG format and I convert it into an ROI assessor, and I see that this DICOM-SEG to ROI assessor is running. And then once it's done, this DICOM segmentation object will be uploaded as an XNAT assessor. That will help me to visualize this segmentation in the XNAT-OHIF viewer. So I see that under assessments an ROI collection is produced, which has the name, which has these 3 different classes: edema, enhancing lesion, and necrosis. So now let's look in the XNAT-OHIF viewer how we can load up this DICOM-SEG object on our scan. Looking at the left panel, we see the different scans, and we see the small icon here which shows me that this DICOM-SEG object is associated with this scan number 9. Now we go through this scan slice by slice, and we can see that this is the T1 post contrast scan that was used for segmentation. From this top right masks option, we load up this predicted segmentation. And we can see in the right hand side we have these different options for switching this necrosis, edema, and enhancing lesion classes on and off. Now going back to the repository, just want to show in the documentation I have the details for doing manual refinements inside this XNAT-OHIF viewer, and I have put a link for that one, and you can see in this XNAT page all the details for doing a refinement are mentioned, where you can paint with a brush. That is, if you think that some part of the tumor is missed out, you can draw that using a brush manually, or you can also remove false positives. Here I'm showing a small example where I choose this manual brush, and then I find a slice where I think some correction might be needed. I turn off these other 2 classes and I only keep the necrosis class on. And then what I do is I reduce my brush size a little bit, and just for the purpose of demonstrating this, I draw a few pixels of necrosis here. Now once I'm done drawing, then I'll just load up all the classes back again, and I can export this refined mask with a new name. So in this case I'm just naming it as refined, and if I press the export button, it gets saved as a new mask. So you see that it shows the mask collection was exported successfully. So now if I go back to the session and I go to the assessors, I can see that this new refined mask is saved with a new name. So this is the end of the entire I3CR-WANO processing on a single session. So we can just download these files as a zip file from the session itself. And let's save it as output. Now if we extract this output zip file and if we look inside it, we can see that there are two folders. One is assessors and one is resources. So under resources, we have all the outputs of the steps 1 to 6 of I3CR-WANO, and within assessors we have the DICOM-SEG objects. Now there are two objects because one is the refined one that we just created and the one is the original predicted segmentation mask in DICOM-SEG object format. So if you want the details of these different outputs that I produced, I have discussed that in my other video where I show the usage of I3CR-WANO using Docker. You can check out that video to see the different output details. So with that, I would like to end the video. But I also want to mention that within XNAT, I showed just a simple workflow where we can go inside one session and launch each individual command manually, one after the other. But there are more sophisticated ways of processing within XNAT. For example, using batch mode where we can launch the commands on thousands of sessions at the same time, or using command orchestration where we don't have to launch these individual commands one after the other manually, but we can just tie them up in a single orchestrated flow. So I will discuss the batch mode and command orchestration in different videos. Until then, thank you for watching.